All right, so what we're going to talk about, we're going to start off, we're going to finish up talking about photosynthesis and some various details here today. I want to remind you that Rubisco catalyzes this first step here. Okay, so it's a really important step. It's where we're going from 3, 5 carbon intermediates to 6, 3 carbon intermediates. You're taking in 3 CO2 to do that, and in between you're adding 1 carbon to each of those 5 to make a 6, and then you break each 6 in half. Okay, that's what Rubisco does. And so Rubisco is important in the regulation of the dark reactions. So regulation of Rubisco is important in regulation of the dark reactions. It's a good place to stop them. Okay, and so let's remind ourselves of what's going on in terms of the chloroplast. So let's actually make it huge so we can actually see what's going on with one of these thylakoids. So what we've got right here is we have our light reactions of photosynthesis going on. And the light reactions of photosynthesis are going to be pumping in protons into the membrane, right? Into the thylakoid lumen. And then over here, or really right next to it, but we're not, I want to make some room here, we would then have the protons would come back out and they'd come through an ATP synthase. Oh, I have your quizzes. Remind me to stop near the end. But they come through an ATP synthase, and then we would generate ATP here. Okay, and that's out here in the stroma. Okay, so given this is going on, right? When we think about this, these come back out, but if they're going to come back out and the energy is going to be used to make ATP, are you going to, as a running rate, are you going to have more H plus out here or H plus out here? Thylakoid lumen, you could just say lumen, or stroma has more H plus. Lumen, right? Because this gradient, H plus coming through, is being used to make ATP. If it was going uphill, you couldn't use it to make ATP. Does that make sense? Mm-hmm. Okay, so we've got more H plus in lumen, and in fact, this is for the mitochondria. I mentioned this. The mitochondria, the big difference is actually the potential across the membrane. Okay, in the chloroplast, it's not that. You actually have the pH. Well, here, let me draw it outside because it's just going to make it small. pH 3 units higher. No, sorry, 3 units lower. I could have saved myself by just changing where I was talking about it, but in lumen. So in here, you have 1,000 times, it's 3 pH units, it's the log scale. So you have 1,000 times as much H plus in here as you do out here. All right, so that's really going to drive that reaction. That means it is basic or acidic out here? It's basic out here, right? Okay, that's going to be important for regulating Rubisco. So Rubisco it needs to have a magnesium. I got a picture down here. So Rubisco to work has a magnesium ion right here that's important for its function. Let's see how red goes across this. It does, great. Okay, magnesium 2+ right there. Just like with MTHFS, we have a magnesium, right? That's important for its function. And some of you are looking at either aspartate 124 or aspartate 154. Okay, for that. I think I made some people switch because I didn't have, okay. Tell me which one you had, and actually if yours didn't work and you're replacing it with somebody else's, I'll probably have the other one in the fridge. Right? But in any case, we've got this magnesium that's essential here, okay? And what we need to have is, here's a lysine. What's this? If that's a lysine, this is why you need to know your amino acids.
Wait, what?
This is a lysine.
No.
There's the amine at the end of the lysine. What's that? We've seen this sort of thing before. It's a carbon dioxide attached to it to make a carbamate. Remember the carbamate for hemoglobin? So what we've got is we've got Rubisco. Bridget, you said it like you didn't like it. It's so cool. We can explain it using charges and Le Chatelier's principle. Same thing here. Okay, this is great. I really love this system because it's something where what's going on in the light reactions makes the concentrations of things all be the right thing, right in the right place. That this naturally using Le Chatelier's principle regulates the reaction. So I've got a lysine. Let me put it in its neutral form. I know you guys hate it when I don't have my protons add up, right? My protons aren't balanced. So this is an equilibrium here, and that's going to be an equilibrium with, I'm going to put just an R here because I don't want to, actually maybe I'll go off to the side anyway. But we're going to have a carbamate. Which has a negative charge and can coordinate a magnesium. Coordinate the magnesium that is necessary for this reaction. And then if I'm actually counting my protons, I lost one of those protons came off, right, to make it H+. All right, so let's think about this. When the light reactions are going, Rubisco's in the stroma, right? We're in the stroma. So when the light reactions are going, you want to run the dark reactions because you're making ATP and you're making NADPH is going to be used for the dark reactions. When the light reactions are running, what happens to the pH of the stroma? Let's go back and look at it. The pH increases, the H+ is decrease, right? pH is always confusing that way. But we're pumping protons out of the stroma into the lumen. So the pH is higher out here, right? Which means we have fewer H+. So look at this. It's like magic, except it's natural selection. But if I decrease the amount of H+ in the stroma, I shift this equilibrium this way, making it more likely to put a carbon dioxide, make a carbonate, which can coordinate the magnesium, which will turn Rubisco on. Right? This is active Rubisco. Active.
What about that? What Rubisco is active? Is it just like saying because the CO2 is attached?
It's active because it needs this magnesium. And it can't coordinate the magnesium without this carbamate.
But what does that do to the Rubisco?
Having the magnesium
makes
it able to do its mechanism, which we're not going to go over, but it needs the magnesium to catalyze its reaction. It may be using the magnesium to coordinate things. So here, right, there it's coordinated. Actually, in our crystal structure, this is a substrate, right? This is one of those ribulose, 1, 2, 3, 4, 5. That's one of those ribulose, is it a 1,5 bisphosphate? But it's 2 phosphates. So it looks like the magnesium is necessary to coordinate this. Or hold it in the active site. You guys see the same thing with our MTHFS, right? There's a magnesium in there. It is actually able to make ionic interactions with the negative charges on the phosphates of the ADP, ATP. Yes?
So when the H plus are pumped inside with the light-dependent reaction, it encourages, encourages Rubisco to
become active.
Yes, okay.
Yep, and you can use Le Chatelier's principle to explain that. Okay, if you're going to run the dark reactions, right? We think about this up here. You're going to run the dark reactions. What do you need to have around?
Carbon dioxide.
Carbon dioxide, right. If for some reason you can't get enough carbon dioxide, we'll talk about that. It's too hot out. You can't open up your pores because you lose all your water. All right, that happens with plants, and we'll talk about that in a minute. So if carbon dioxide's low, you don't want Rubisco to be active, right? But if carbon dioxide is high, I'm going to make this all making it active. If you've got your carbon dioxide around, then you should also shift that direction to the carbamate. You make the carbamate, you coordinate the magnesium, and you can run the dark reactions. Does that make sense? So we have two conditions that we want the light reactions running. We also want carbon dioxide around if we want to activate Rubisco. We can explain both Le Chatelier's principle. Questions? Okay. So we have another system where this thing is reacting the way you'd expect based on the things that are being made in its environment. Okay, or the things that are present in its environment. Like we saw with hemoglobin. Questions?
You said that while you're running the light reactions, you're still running the dark reactions?
The dark reactions do not run if the light reactions aren't running. Because the dark reactions use the ATP and the NADPH that are made in the light reactions to run the dark reactions. So if you're not making ATP and NADPH, you're not going to be doing this. That makes sense. That's why it sounds like in biology they call them the light-independent reactions instead of the dark reactions. They don't like the dark reaction term. Okay. So let's go ahead and move on then. So what I'd like to talk about now is we're going to talk about C3 and C4 metabolism for plants, okay? So plants, depending on what's going on, they can either do C3 or C4, so 4-carbon intermediate or a 3-carbon intermediate photosynthesis. Actually, let's talk, before we talk about the one way that it gets used, let's talk about what it is.
Isn't there also the can plants?
What was that?
The can plants?
Yeah.
Okay.
So this, this is a picture of, these are stomata, okay? And so for photosynthesis to occur, these are little pores in leaves. Those pores need to be open to let in carbon dioxide, right? Okay. However, having pores open lets water out. All right. So when it's hot, really hot and dry out, what's going to happen is those pores are going to stay closed to keep the plant from drying out. All right, that makes sense? So what that means is that it could be super, you could have lots of sun outside, but you can't get any carbon dioxide to run photosynthesis. Okay. So essentially what would happen is a light reaction would run, you'd make all your NADP plus into NADPH, you'd make all your ADP into ATP, and then there'd be nothing else to do with it. All right, so some plants have adapted to this by storing carbon dioxide in a 4-carbon intermediate. We're going to call these C4 plants. All right. So we have a separate cycle that we'll talk about. Let's talk about it now, and then we'll talk about a fun way we use it as humans. So we've got C4 plants. So what does that look like? You won't need to know this. You need to know the general bits about why plants do this. Okay. So this is what the whole pathway looks like. All right, overall, and I'm going to blow this up so we can talk about bits of it. All right, so what can happen is you can take phosphoenolpyruvate and you can use phosphoenolpyruvate carboxylase to make oxaloacetate. Have we seen that reaction before? Yeah, where do we see that?
Gluconeogenesis.
Gluconeogenesis, right? We're going to make, so we can do that. We can make oxaloacetate, and then we can use malate we can use some NADPH, right? Do we have a bunch of NA? Well, if light reactions are running, we have NADPH around, right? But not necessarily when it's dark out. But when it's cooler, you can go ahead and take that NADPH and use it to make a malate. All right, and this is how we're going to store. We're storing in here. We're storing that carbon dioxide as a 4-carbon intermediate. Then, when you want to use that carbon dioxide in the Calvin cycle, what you can do is you can then get your NADPH back and break that off as carbon dioxide and generate pyruvate. All right? So we used an NADPH for this, and we got the NADPH back. All right? So in doing that, did we use any energy? In terms of the cofactors, we used it and got it back. However, where did we start with this process? Was it pyruvate? So we used phosphoenolpyruvate up here, right? So we didn't need to use ATP there because we weren't going from pyruvate actually. So we haven't exactly seen this enzyme, this reaction before. But we started with phosphoenolpyruvate, right? And we got pyruvate out the end, right? But if we want to go from, in gluconeogenesis, how many ATP did it take to go from pyruvate to phosphoenolpyruvate?
1 ATP and 1 GTP.
1 ATP and 1 GTP. Do you guys remember that sort of from the bottom? Do you want me to go back?
Yeah.
Well, let's do it.
Is it like, would you have to make 2 phosphoenolpyruvates to like go all the way back? So would you need to use it twice technically?
I don't think so.
Cool.
All right, so we're talking about this, right? So we start that C4 process here. We make oxaloacetate without taking energy because we're going from here to here. Okay, not using that. But we make oxaloacetate. Doesn't look like we can get ATP out of it, which is sad. But we make oxaloacetate, and then in the end we end up with pyruvate. So then the plant needs to regenerate that phosphoenolpyruvate, and that's going to cost 2 ATP. So it's costing 2 ATP to skip that CO2, right? How many CO2s do we need per Kelvin cycle? 3, yes. Very good, because we're making a 3-carbon intermediate that we're going to pull off a 3-carbon intermediate. Let's go back to our other thing. So in terms of this regeneration here, right, from phosphoenolpyruvate to pyruvate, that's 2 ATP to go back to make phosphoenolpyruvate again. You guys with me for that? And then if we go up to our Kelvin cycle, we need 3 carbon dioxide, so 6 ATP, right? So this process without C4, this is just C3 right here. Okay, C4 is an extra process, and you still use the Kelvin cycle. But we've got 6 ATP, 3 ATP, right? One cycle is 6 ATP. No, I'm sorry, 9 ATP. It's 6 NADPH, right? And then with C4, we've got another 6 ATP, right? 2 per carbon dioxide we have to spend. You see that running C4 is more expensive? Okay, so running C4 is more expensive, but it's worth it if you live in a hot and dry environment, you being the plant. Okay, because during the day, you can still be running photosynthesis when the plants that can't do C4 can't. Does that make sense?
So it's 6 extra.
6 extra ATP, and that's in the regeneration of that phosphoenolpyruvate. So let me go ahead and write that. I'm not going to write it out with the numbers, but I'm going to say, so the C4 cycle, or the C4 process, I guess I should call it. It's not exactly a cycle, but the C4 process is expensive, but worth it in hot and dry environments. Because C4 plants can be running photosynthesis when C3 plants cannot. Why can't the C3 plants do it? Because they can't open their pores because all the water will go out. Okay? Questions on that? Okay, so I don't expect you to know this process. We walked through the energy bit. I don't expect you to be able to explain the energy bit, but you do need to tell me that what's going on in C4 is carbon dioxide is stored in a 4-carbon intermediate, and that it's more expensive. Okay, and then be able to talk about why these plants will do this in terms of they can't let their pores open because they'll dry out. Questions? Okay, let's talk about a fun way that this gets used. So C4 and C3 plants incorporate carbon-13 at different rates. Let's say at different ratios to carbon-12. Okay, so you're going to get a different C13, C12 ratio. You guys remember from NMR, you'd have your C13 peak, right? Okay, because C13 it's about 1% abundance. It's not horribly abundant, but it is abundant. And so you could pick that up on mass spec. Okay, and so each of us eats a different ratio of either C3 and C4 plants, or eats animals that ate a different ratio of C4, C3 plants. So each of us eats a different diet that results in a different ratio of C3, C4 plants consumed. And that's either directly or indirectly. So if the animals you eat ate plants, it goes back to plants.
What's the difference that they do in their bodies?
There's not really a difference that I know of. Okay, but you can detect kind of these ratios. Okay, so these ratios show up in your hormones. Including testosterone. So what's going to go on here is this ends up being, you can use, this is the gold test for steroid use. So in the Olympics, what you can do is you can test whether somebody's steroids are hot. Their testosterone is high, right? Their natural testosterone is too high. But of course, Olympic athletes are going to have high testosterone anyway, male or female, because they're they're Olympic athletes. So how do you know whether it's the real testosterone or they're taking testosterone? Well, the testosterone in their body will have a certain ratio of these things, right? And then their other hormones, you can look at their other steroid hormones, and that ratio should match. And if it doesn't match, what it means is that you're taking some sort of synthesized testosterone, which is going to have a different ratio of C4 to C3 than you. Does that make sense? Okay. And so what you do is you do a mass spec of that sort of thing. I'm not going to ask you about this. Maybe I won't write the rest of it out. And I've had people ask me before, well, couldn't you fake your testosterone and have it match? I'm assuming that's prohibitively expensive or difficult to make that happen. But that's probably what's going on there. Questions? Okay, well, that's all I have before the exam. Do you guys have any questions you want to ask? Or we haven't prepared for our review day yet.
I have just one question.
Yeah.
Okay, so like, there's when we were doing like the different like electron transport chain
Yes.
examples, and one of them was like pyruvate or something.
Yes.
And then the last was like another.
Yes, for the quiz. Well, stand back the quiz.
Why did we? It was like, I don't know, we did like the first one and then the middle one instead of the first and the last, 'cause I thought you said that if there's a two-way arrow, like for the, I don't know, one, like yeah, one, like you didn't, it would like cancel out. You just can't do it. Cause like you just had the one for like the normal electron transport, and then there was an example one,
Right.
and it was not asking me to find like the overall.
It was, I think it was.
When I was studying for like one of the
It
quizzes,
was
I
like here, and then
Mhm.
like why did you do basically this instead of this? Like wouldn't these cancel out?
Oh, well, we can do, okay, so the question, good question. The question is, why aren't we looking at one step of the electron transport chain as opposed to one side to the other, right? I mean, they are going to cancel out, but, so for the quiz, did I even call it a reaction? Okay, so I took just one step, but you can calculate the reaction for any given step, right? As long as you make a full reaction.
So that was okay. But the question was making ETC using pyruvate to generate the Marshall gas spontaneous at each step. So then I thought we would be calculating number of times.
The one that we did in class?
Yeah.
As opposed to doing just one step?
Yeah, like I thought this question was asking to make it spontaneous and then we calculated energies to make it all spontaneous.
Yes. So you could do that.
But that's.
But you have to calculate, to do that, you have to calculate. I was only asking for 1/3 of that.
Yeah, but I was just asking in this example, we were just confused because like the question that I written as a prompt was for the whole thing and then our like energy calculation was one part of it. Like it was like the one step where we got the whole thing.
I'm trying to, I'm passing these out, sort of curious from science, and I'm trying to understand. Let me finish handing these out. I can talk about, I mean, so what I'm testing here is that this is about that you know how to do electrochemical reactions, you know how to calculate Woodward's half reactions. The higher concept is, you know, also knowing what's spontaneous.
I was also thinking micro and technically Holland in one day. I'm proud of myself for that because I did not think I was.
That one little part.
Okay, so I don't know, have I answered the question or not?
So, okay, basically.
So the concern, the confusion is, why did I do the quiz the way I did? Okay.
For this example.
Yeah.
Well, in my notes, I have it as for the whole reaction.
Yeah,
And then the
we did do that.
example is for one step. Yeah. So it was just like a misinterpretation.
Okay, so we're back, was it this day?
Yeah.
Yes.
Okay, so we're looking at this step, was it this
No,
example?
it was like the whole thing. It's after that. It's after. Yeah, right there.
Okay,
So then it's like, why, like, this should be calculated for the whole complex. So why are we only doing the first?
I thought it would be lactic to pyruvate and then NO3 minus to NO2. And then
well, this is not, what we've got here, the lactate to pyruvate, we're just calculating for this step here, we're not doing the full reaction.
Yes, okay, that was just, okay. That was
And so this was make an electron that's spontaneous each step, and then I may have just been showing you how we do it where we don't just use the ends. Because if I just use the ends, then you wouldn't ever look for this quiz. You would have put all the notification on that. Okay? But you can calculate the potential change for this and for that, right? But if somebody asks you for the whole thing, you just need to use that. You just need to use the front and the end, because this one is going to be it's going to be positive whatever it is in one direction and negative whatever it is in the other direction, if that makes sense.
Yeah.
Well, it's going to be this half, and it's going to be here to here and here to here. And they're going to, the fumarate succinate parts are going to cancel out.
So, for the exam, I guess it's just like just everything we've done.
Yes,
Okay. So,
it's a lot.
yeah.
So you have 2 practice exams that are up. Take a look at those. That'll give you, an idea of kind of what sorts of things are going to happen. I mean, in terms of emphasis, you have the study guides, but step in glycolysis, step in the citric acid cycle. We've got the pyruvate dehydrogenase complex. I don't think I ever got a quiz to ask you about fructose 2,6 or 2,6 bisphosphate, right? Regulation, but that's important. Okay. So do you want to review that real quick?
Yeah.
Yeah.
It seems like that would be a good one because we didn't get a quiz on it. Not too far. I just went past it. Okay, this is it. All right, so first off, here's where we start a regulation. What we were doing is we were stepping back a couple steps from this, and then we went to a miracle occurs, insulin binds to the membrane, stuff happens, there's a bunch of steps, and then we picked it up right near the end. But this is the very end, right, in terms of regulating phosphofructokinase 1 and fructose bisphosphatase 1. But just a general question here, why this step? Why regulate here as opposed to someplace else?
Is it commitment step?
Well, yes, why is it the commitment step though?
So you use ATP.
After that, all the intermediates are exclusive to glycolysis.
Okay, so after this, things are going to go to glycolysis, but do you remember that glucose 6-phosphate? This is where we branch off to go to make glycogen, or we'll branch off to go in the pentose phosphate pathway. So we've got a lot of stuff that happens here, comes off of here. So we don't want to really regulate, I mean, there is regulation here, but we don't want to regulate hard before that because we want this made, as we could store it, or we could use it for energy, or we could use it to, there's a bunch of different things we could use it for. Okay, so regulation, there is regulation, all these sorts of steps, but this is where the real regulation is for glycolysis, because this can go to a bunch of different things. Why not regulate this?
Double arrow.
It's a double arrow, it's reversible, okay? So if this has one enzyme for both glucose and gluconeogenesis, or glycolysis and gluconeogenesis, it's shared. So if you turn that off, you turn both processes off. You want to be able to turn one on and turn the other one off. All right. So we need our single arrows. We want to have it after we branch off to a bunch of other processes. So this is why we're here.
So because I can just go a bunch of different ways.
Up here, yes. We don't want to regulate. We want our commitment step after we make a bunch of other things. I just lose everything.
So it's the step after we make a bunch of other things.
But after you branch off to a bunch of other things, right? Is it here? Yeah, there we go. Okay, so let's start from this side and see how that works. All right. So back here, back here. Okay. This is back a number of steps. So we have insulin can bind to the outside of the cell, binds to a receptor on the membrane, and we end up with a bunch of signals. A number of steps that happen. That's what these dash. Or glucagon can bind to the cell. Okay, one of those is going to bind to the outside of the cell, and it's going to send a signal in. And after a number of steps, we get down to where we have our phosphofructokinase 2, or FBVase 2. Okay, fructose bisphosphatase 2. We have there. It's a combined enzyme. Okay, which you don't need to. I don't care whether you know it's a combined enzyme or not. But what I do care, is that if you take the phosphate off of it, or off of whichever one you want to talk about, insulin signals that taking a phosphate off, when we take a phosphate off, we get a shape that has either change in shape that gives us phosphofructokinase 2 being active, and no phosphate means that fructose bisphosphatase 2 is inactive. All right, this enzyme is going to make fructose 2,6 bisphosphate, which is the signal molecule for up above. Okay, way up at the top. If instead we have glucagon, what we do is we're going to go ahead and phosphorylate this thing and cause a change to happen. All this stuff, this is not rational. You rationalize what needs to happen for the full process based on insulin and glucagon. You can do that because they signal certain things. But whether this reaction happens or not is not a Le Chatelier's principle thing. It's not a state of the cell that I have lots of ATP around, so I drive this reaction. None of that stuff. This is pure signaling mechanism. Okay, does that make sense? So we're not gonna. So now let's say we have insulin outside the cell. We've activated the phosphofructokinase 2. Phosphofructokinase 2 is active, so it's going to take fructose 6-phosphate, right? We're making that in glycolysis, and we're going to make fructose 2,6 bisphosphate out of it. Okay? So we're making this because insulin bound to the outside of the cell, and then one more step up, fructose 2,6 bisphosphate will activate phosphofructokinase 1 and cause glycolysis to happen, and it will turn off fructose bisphosphatase 1 and turn off gluconeogenesis. Okay? So in the liver, liver's a great cell for this because the liver does both of these processes, right? It does glycolysis and gluconeogenesis. What goes on is, so insulin binds the outside of the liver, and what it's saying is, hey, insulin signals you have high blood sugar, right? Insulin binds the outside of the liver cell. It says, hey, there's lots of glucose around liver, so it's okay for you to use it for energy. Okay? Also, what's going to be going on is the liver's going to be making glycogen. But it can make glycogen, it can use glucose for energy, it's not in short supply. Okay? If glucagon binds to the outside of the cell, go down to the bottom of that process, what we have is we phosphorylate this enzyme complex. It's going to break down fructose 2,6 bisphosphate, right? This guy's active, we're breaking it down. And what we get, we stop inhibiting fructose bisphosphatase 1, and we stop activating phosphofructokinase. So now this process is active, and the liver is going to start making glucose from pyruvate, so it can dump that glucose out into the blood for the other tissues. Okay, so like for your brain. And we'll learn a little bit later what it can do is you get when you break down your proteins, you get alanine gets put into your blood, which then gets turned into pyruvate, which then gets made back into glucose by your liver. Your muscles, when they break down stuff, they don't build it. They don't make the materials to build it back up. They're waiting for the liver to hand them glucose. Yes.
Why do we need that intermediate step? Why don't insulin and glucagon just regulate phosphofructokinase 1 and fructose bisphosphatase 1?
There's a enzyme. There's a cascade here.
Mm-hmm.
So you've got multiple things where you're amplifying from the signal from the outside of the cell, and you see that this is an amplification step, right?
Mm-hmm.
So what we do is if we activate this enzyme, we make lots of these. If we activate this enzyme, we tear down the lots of those. So it's one of the amplification steps in the process. Other questions on that? So that's not everything, but go ahead, look over the study guides, look over the old tests, and Wednesday come in with questions about these things or things you'd like to review. All right. You guys have a good day. Do come in and do your